Well, first of all, let me thank Jacques, Antoine for the kind invitation. I'm, just like Jan, I'm very honored to speak in this in front of this audience. And I'd like to tell you about how we use single molecules and super-resolution microscopy to address what I call, in situ biochemistry, or in other words, how you can, measure and biochemical interaction by photonic means directly in the cell. So, of course, you'll see there are several arguments I've heard before, and I think I definitely agree with a lot of the things that were put forward by the previous speakers. But I'll try to give you my perspective, maybe with a little bit of the biophysics background. So, as just one introductory slide, I wanted to start with an historical note and coming back to the early days of biological imaging and microscopy. So, it's known that it dates back to von Leeuwenhoek, and I truly apologize for the improper pronunciation of the scientist. But the important thing, I think, is here that it dates back to about 350 years. So it's right at the time of this Academy of Science was founded. And actually, I think he soon after became one of the, corresponding foreign member. So, and of course, with this microscope that you see here, the early days, it could already report observation of microorganisms. And so it was a big step forward. And if we just move along in the 19th century with a better understanding of optics and building of better optical instruments, probably exemplified by Ernst Abbe, which is the founder of one of the founder of the microscopy industry, as well as being a scientist, of course. Using microscope now working very close to the diffraction limit, I think it led to the discovery of subcellular organelles. So each time I think there was progress. I think there was opening a new window on the biological world and leading to discoveries. So now, 150 years later, we're here with the development of super-resolution optical instruments. And I think it was already exemplified in the previous talk, but I think it clearly opens exciting prospect for investigating biological structures at a truly molecular scale, and that's, of course, what I like to emphasize in the rest of this talk. So the tool, and I don't need to spend much time, but I think it has been explained by the previous speakers. The main tool we're going to use is this single molecule localization microscopy, meaning the ability to look at a single fluorophore, single fluorescent protein, single dye, and to image it on the camera and to localize it by recording the fluorescent signal and finding the exact position, or at least exact with some nanometer accuracy. And now if you have something with moving like this little protein in the membrane, then you go from imaging to set of localization to reconstructing trajectories, so you have a tracking with, again, nanometric resolution, but in temporal resolution up to about 1,000 hertz, like a millisecond in the best conditions. So clearly you're able to explore a biological environment at really the nanometer scale, and that's going to be one of the main advantage that we're going to take benefit of. So to tell you about I think is it's going to be very useful for. Just let me make a very general comment about this property of supramolecular assemblies in cells. So I took 3 examples. They are total, kind of arbitrary, and definitely non-exhaustive. I took a schematic of the transcription machinery, the membrane nanodomain, the so-called lipid raft, or neuronal synapse, and they all represent what I have in mind when I talk about supramolecular assembly. So these are structures, usually sometimes self-organizing structures, and they have this specific property that make them really hard to look at by a conventional means. There's these structures of a size on the order of 1 to 100 nanometer range, and I realize now that the nuclear pore that was described before is another beautiful example of this assembly. So the size in this sub-diffraction range, the components are present in a low copy number, like 1 to 100, so it's not easy to measure by standard biochemical means. And on top of that, their individual components are constantly and rapidly exchanged. So things go in and out, but the entire structure remains functional and relatively stable. So we have to deal with this type of constraint when we want to measure them. And I think has been a limit to look at them by standard purification method or looking in a biochemistry. It's not impossible, but it's really clearly challenging. And if now you look at it from the perspective of single molecule measurements, clearly you can think about addressing the composition of structures, because you can count individual molecules. You can track individual molecules, and you look at them like moving in the cell. And of course, as described before, you can look at the structures by the standard, not the standard, but the recent palm storm microscopy methods that have been presented. So really, you have a way to address some of the most challenging aspects of this at this supramolecular scales. And I think in the rest of the talk, I would like to illustrate this in a specific example by stressing maybe some of the biophysical questions we had in mind when we started using them and addressing them, and where we are right now. So the story, and maybe I'll ask it to be, okay, with the dim lights, without the chandelier, and we can keep it like this maybe for the rest of the talk. It started like many years ago, and I should say that's really, it was thanks to many discussion and exchange and work with a group of Antoine Triller at the biology department of École Normale. So this was the question of the origin of the neuronal membrane, and that's really what Antoine convinced me that we should team up to look at. And you see in these beautiful images like a set of like green and red dots, and they correspond to inhibitory and excitatory synapses. And clearly, when you look at this, there's a question of how these thousands of sites are properly organized. They just contain the proper set of molecules, and how possibly they are regulated during different type of activity or during the synaptogenesis. So to do that, and that's now kind of started like many years ago, was to look at it from a single molecule perspective, to try to be able to track receptors, like glycine receptors going to inhibitory synapse, interacting with a scaffold, the GlyR-gephyrin scaffold, and to look at it in live neurons. And this is really one movie that I think captures the most of the important messages that we were able to get. That you see here a dendrite. And here it is a synaptic site labeled in red, and you see these little receptors which is like moving around and then they're trapped right now, staying at the synapse and going on, and then you can repeat the movie. So I think the important message we got at the time that not that receptors move in the membrane, probably that was something we could expect. But I think more importantly that the interaction here at this site, this little scaffold, with the scaffold, these GlyR-gephyrin clusters, is relatively transient. And for example, if you plot this type of data in a graph where you go in and out, so it's very analogous to the kind of data you could imagine in electrophysiology, where you have a channel which is open or closed, just because it's just a two-state system, you can have this kind of dynamics which occurs on the level of a few seconds. So these receptors get there, but are only transiently stabilized. So the picture we had, and it was from maybe just maybe a little bit naive physical perspective, but that these sites, they act as some sort of a trap where receptors by diffusion get there, so they get trapped. But importantly, this is not a very deep trap, and the relatively shallow trap depth, make it possible for them to escape. And that's the picture we had in mind, but it took a long time to get to get to a quantitative description of these, and that's what I'd like to show you now. is that how we can measure the property of these traps, quote unquote, at the single synapse level, and also maybe wonder whether these properties, the size, the amplitude of the binding, some sort of maybe mobility properties are regulated. And that's the question we had in mind when we started. So the first thing, and so I can really use now all the arsenal of tools of this single molecule imaging, was to try to measure numbers. So you had this, and the way we did it was a really, in a sense, a very simple, but it turned out to be a relatively efficient way, was to record the intensity of these spots. So you see individual spots correspond to GlyR-gephyrin clusters, so synapses expressed in a knock-in mouse. And of course, if you shine light, as we know from biological imaging, everything gets dark after a few seconds or maybe a few minutes because we bleach everything. So we start from frame 1 and we go after a few frames, we have like nothing, just background. But a property of this fluorescent protein, I think in this case it's RFP or GFP, I think it's RFP, is that they are not entirely, they don't necessarily turn off completely permanently. They have some sort of revival. So they come back and they emit a little bit of light. And so from this, we can try to estimate the signal of one molecules and doing some sort of extrapolation, evaluate the initial number of molecules. So that's relatively simple in principle. It needs a little bit of a careful analysis, but it turned out, and we cross-check with other methods, that's not a bad way of doing it. And then we could estimate the number of GlyR-gephyrin molecules. So in a single synapse, in one cluster, and we measure endogenous MRFP GlyR-gephyrin, we have about 200 protein at a single synapse, with obviously some sort of distribution. All synapses are not alike, but this is the order of magnitude. So if you want now to one more point. So of course, the question was to say can we correlate the number of receptor, or at least receptor binding sites, with the number of GlyR-gephyrin molecules. So we could try to use the same thing, but now extend it to 2 colors. So we have this scaffold tagged with a red protein. The receptors was tagged with a blue protein, in this case a photoactivated protein, but the point that we can measure them independently. And I should say in this case, it's not completely a receptor. It's a transmembrane domain that binds to the scaffold. So it's, in a sense, measuring the number of binding sites. And then we do the measurement in non-neural cells, so we don't have to compete with endogenous receptor or endogenous GlyR-gephyrin molecules, and then correlate these two numbers at the single cluster level. And what the pictures we get from there is that they seem to, very linearly with one another, suggesting that there is always some sort of the same number of sites available, and that in this case we think because we overexpress this protein, we saturate these sites. But the fact that it changed linearly means that if we change the number of binding sites in red, well, it seems like we always get the same number of bound molecules. So it's something that was important information from there, is that it supports the idea of a clusters of a scaffold as a two-dimensional organization, possibly a lattice organization, as suggested by people looking at the structure of GlyR-gephyrin and the oligomerization domain of the protein. So maybe it's hexagonal like this, but that we have no proof of that organization. But it suggests really a two-dimensional clusters where receptors, or at least the domain containing the transmembrane protein containing the binding domain can attach. So when we were there, as we were getting more and more information about the structures, we wanted, of course, determine some sort of a dimension of this object, and then we started doing that we had this emergence of superposition methods. So we try to look at this now with a type of pound measurement. So we really reconstruct the organization of the cluster and measure the size. But something which was a challenge at the time for us is that, of course, the clusters is not a synapse is not necessarily parallel to the glass cover slip on which the neurons were plated. So we had an angle possibly along the, certainly, if you look at imagine the tubular structures of a dendrite, you can be on the side. So clearly we have to measure in 3D. So, that's where we play a bit of optics. And because we had to face the classic problems, I will summarize in one slide, that if you look classically at the signal of a fluorophores in 3D, if you're in and out of focus, you have this kind of, you expand the range of the spot, but more importantly, it's symmetric if you're with respect to the focal plane, meaning you have the same size if you're above or below. So it's hard to get a good Z position from just an image like this. So we had to play a little trick, which consisted in using a deformable mirror. So that's an idea coming from astronomy, where they use adaptive optics to image the stars and galaxies. In this case, it's a much smaller scale, but the same techniques can be used to do what's so-called astigmatic imaging. So instead of breaking the symmetry along the z-axis, and then by looking, so you have this weird astigmatic PSF for the optical physicists in the audience, but you can calibrate that and get a relatively good Z position, not as good as what was shown before by Jan Ellenberg, not 10 nanometer, but more like 40, 50 nanometer, which was in a sense sufficient for us to get a first view of a super resolution of this synapse, where you have in blue the receptors, which are labeled in this case with an antibody, and also the gephyrin clusters. And actually the space that you see between these two structures very much relate to the fact that the label on the receptor size is displaced by the size of this antibody. But nevertheless, we could kind of characterize the size of this domain, giving you an idea on the order of 200 nanometer, typically. Of course, a lot of variability. No synapses are alike. And so leading to a density of scaffolding molecules, since we can count them and we can count the size, on the order of about 5,000 per micrometer square, which is relatively dense. So again, if there's some sort of lattice organization, there's no proof of this, direct proof, but at least it shows that it's relatively dense. So if I now summarize this part about the nanoscopy of this so-called quantitative nanoscopy of inhibitory synapses, we have now a lot of numbers to tell us about these structures. And so size, count number of quantum molecules, maybe number of binding sites, and so on and so forth. But clearly the things we had in mind was to measure also the kind of the binding energy, the strength of the adhesion, the interaction between the receptors and the clusters. Why, what that controls the stability of the receptors at the synapses. So it took us a while just to find the right way of doing it, but it came finally with the emergence of the methods which are intermediate between the palm storm super resolution and more the tracking that I showed before, where you see only a single spot, which are based on the so-called SPT palm, so single particle palm, and which is again based on the stochastic photoactivation of the marker. So you turn a non-fluorescent protein into a fluorescent one, and then you follow the motion. When it bleaches, you turn another set, and you repeat the measurement. Another way of doing this, just for aficionados of single molecule imaging, is that you can use antibodies. These antibodies, you put them in solution, they diffuse, they bind, then they move with the protein along the membrane, and either they detach or they photo-bleach, and other will come. And if you tune your concentration of antibodies properly, you can achieve a nice result. It's a technique developed by Gregory Janon and collaborators in Bordeaux. And that's actually a very powerful method to track membrane receptors. So the data that you get with that, they look probably very similar to what you saw before. So you have these individual spots. They come, they don't stay very long because the proteins are not very photostable, but you see that they move. In this case, the dye, you have another type of label, so they are a little bit more stable. The signal to noise is higher. You look at them for a long time. But the real message for us, what was important for us from this type of method compared to this tracking that we had done with Antoine earlier, is not that we have that we can achieve a large amount of data, and So instead of looking at 30 to 100 individual trajectories per conditions, like we did in our first paper, like a few years ago, now we can really achieve millions of individual detection in a few minutes. So maybe in the era of Google and Facebook, we cannot call that really big data because we're far from the gazillion terabyte that they record. But still, from a biophysics perspective, it's a lot of trajectories, a lot of detection. And they provide a lot of information. So we can actually now think about how using them. So the computational methods, I think, are becoming more and more important, and I will try to see, in our case, how we try to take advantage of this large amount of data. And it was on the Bayesian inference methods, which are relatively standard for people doing statistics or applied math, maybe a little bit less common in the field of microscopy, although now with the ever-increasing amount of images and data that we acquire, it's becoming more and more frequently used. So in our case, how does that work? So you have all these points that are moving, and I think you should think about these individual proteins moving in the membrane. These are the trajectories reconstructed from the data. As little explorer, they move in the membrane or in their cellular environment based on some property of the local environment. And that we like to extract, to infer. So the way we do it is to use some physical model with we have like noise, like the diffusion, which is driven by some thermal noise, but you have also some transport or trapping. In this case, we think about the clusters as a little trap. So we use some sort of an effective potential description, and we like to infer that. And if you're familiar with Bayesian inference, it's going to be very trivial. If you're not, I try to just give the idea in a simple words. If you look locally, I mean, you have plenty of molecules going through this little part of region of space, and so they give you information about the motion. But this motion is driven by some models. So what we compute is the probability of the parameters given the data, and which, as is known from Bayes' law, is proportional to the probability of the trajectories given the parameters times some prior information. So just to get to the important message, I think, in our application, is that with this we try to distinguish between the contribution to the motion. It can be in heterogeneity in the diffusion part, where you have parts which are more viscous than others, but proteins are not necessarily trapped. And from the movie, we can map that, but we can also have really a real trap in a sense of a place where a protein will fall and move around, but before it can escape. But this is a different. We see that as different processes, and so that's what we used now in the context of the interaction between gephyrin and the glycine receptors. And again, we did not use the glycine receptors. We used a transmembrane domain that possessed the loop, which is known to mediate the interaction between the receptors and gephyrin. So this binding region, which is here, was fused by collaborators in Antoine's lab, Christian Specht, to a transmembrane domain with a probe, and then we can start taking the movie again. So again, same type of movie of all these spots correspond to receptors, this transmembrane domain, exploring the environment. We track them, and we try then to map entire. So this is a kind of the physical view of what for that specific transmembrane domain, given these interactions, what in a sense the membrane look like, where you have something which are heterogeneous, both in terms of diffusivity and some sort of what this effective potential that we try to infer. It's very clearly seen when we plot this as more the force, the trapping here, and you see this site. And if you now look more carefully, you see this spot, and they correspond to the gephyrin clusters that really act as a trap. So this is infer from data, and then we can try to estimate the depth, so the stability of this area, in a sense of expressed as the depth, these parameters of energy. So we have an access with this type of approach, we think to the amplitude of interaction between receptors and the gephyrin. So if we did this measurement for different structures, so we will use it for for different binding loop, the wild type, but some where we know that there's no interaction, and some intermediate one, which corresponds to the action of a phosphorylation, which is known to regulate the interaction between the clusters and the scaffold and the receptors. In this case, we permanently modify the amino acid, and then we do the same experiment with all these constructs separately and try to infer for all of them the properties. And clearly, it's as a first step towards studying the regulation of this binding energy, this stability. We see that we go from something which is more stable. We reduce the stability by changing this loop. But we never get to very high energy on the order of like a few kT, 5 kT, maybe 4 or 5 for the wild type, to something which is almost non-significant in terms it's 1 kT. So we think it should be almost considered as 0. But this is really our approach to extracting some sort of real biochemical information by using this sampling coming from high resolution single molecule data. So I think I'm getting close to just the final slides, because now if I come back to this little picture, that was of course a simplified view of what would be a synapse, but at least from let's say a biophysical perspective, it's not unrealistic that we have this trap, this coefficient, this motion, mobility in and out, and we can extract all these parameters. And we hope that, I mentioned them, how fast they move, the size, the density, and the stability. And I think with this, we can now really correlate that with probably regulatory processes. Oh, thank you, Google Drive. Excuse me. And importantly, it's something we haven't really used so far too much, is that it's measured at the single synapse level. And we'll see clearly there is a big variability across just a single neurons. And so there is a way just now to really look individually, to look how some of all these parameters are correlated together, and maybe to get a good structural but also dynamical description of these synaptic assemblies. So I think I'll conclude and give just maybe a short perspectives. That really, I think the message, again, I think it goes along with what other speakers said, that the single molecule tools, they provide access to structural composition dynamics of assemblies in their natural cellular habitat. And that's, I think, the new step, and that can be used to address many, many questions. I like to stress that, of course, there are going to be some sort of future optical methods, and I think we're not done with developing microscopes in particular for measuring 3D dynamics. But if I were to predict something, I think it's going to be the increasingly important role of computational methods. I think Jan Ellenberg showed how the single particle averaging was actually very powerful to reconstruct the structure of nuclear pore. I tried to give some hint about the use of statistical Bayesian methods, but I think there is a really almost an untapped potential in all the tools that comes from applied math and statistics, in a sense that really a way to describe this data, and also this concept of machine learning that are now almost everywhere. I think they can be extremely useful because as we get with more data, we have to classify them. We know that everything seems to be so heterogeneous, so we need a proper way of classifying, making proper categories. And the more data we have, the easier it's going to be. And clearly, these tools coming from so-called big data and all this computational aspect, they are actually designed to do that. So completely sure we'll see more of these in the near future, applied to biological single molecule and super-resolution images. One of the messages about this interaction with different clusters and glycine receptors is the role of weak and transient interaction in the subcellular architectures. Clearly, a lot of this the association between protein partners and molecular partners, they don't have large affinity. They are sometimes hard to capture in standard biochemistry. We see them so constantly in single molecule images that, and we see more and more evidence that they actually play a role and they are part of the system. So that's, so these are not spurious interactions. They are really, in a sense, intrinsically weak. And finally, of course, there are a lot of challenges. We try to bridge gap between different scales, going from the molecules to the cells with this intermediate supramolecular level. But we have information from in vitro data, we have from ensemble data for the population level, and we have this intermediate value which comes from analysis in cell, in single molecule in cells. And in this particular case of synapses, of clusters, of different clusters interacting with receptors, but in many cases, I think this is a kind of a challenge to really reconcile all this information to get more comprehensive and quantitative and possibly descriptive description of our systems of interest. So with this general comment, I think I need to stop, but not forgetting to thank some of the people who do the work. So in this particular case, I really like to stress the role of Ignacio Izzedine, Bassam Aj, and Mohamed El-Berri, and also Mathieu Copé. But in my group, this was all this Bayesian inference techniques was really through coming from discussion and also from the work of Jean-Baptiste Masson, who is now a group leader at Pasteur. And I mentioned, but I think I should stress it again, collaboration with Antoine and people from his lab, in particular recently Christian Specht and Charlotte Salvatico. And I think with this, I'm going to stop and thank you for your time and attention.
All right, any questions? Oui. Allez-y.
The analysis in terms of a kind of energy well with the width and the depth is very elegant. Do you have an idea about it going from one synapse to another, whether you would expect, if it's a stronger interaction, that you would have a deeper well, or would it be a wider distribution?
I'm sorry, maybe I missed a bit your question.
You
if you go
your parameters L
yeah.
and U.
Yeah.
So if you're going from one case to another, and the second case you think is a stronger interaction, I mean, how would you expect it to vary? Would you expect the L to get bigger or the U to get bigger if you're going from a small synapse to a large synapse, for example?
I think very often, I mean, we see, well, we see very often that we get larger. We have a distribution in size, but very often there's no clear correlation between the size and the depth. And I think it reflects, in a sense, this kind of the fact that it's somehow we think it's kind of two-dimensional structures. So the trap, is not something which is going deeper like this, but more kind of extending. So we populate a bit more, but it's not necessarily more stable. Of course, there are variability, but there is not clear correlation in one sense. This being said, we see that some, depending on the regulation, if you modify the binding thing, clearly it changes the depth. So in a sense, the stability at a single synapse. So I think, but these are two different type of regulation.
Yes, Daniel Schuke.
Yep. Yeah, another view of these potential wells is the affinities of the interactions, of course. And so one parameter you have in your data is the residency time of the immobilization of the trajectories, in the trajectory. So I was wondering whether you actually compared the affinities you could infer from the residency times, from the arrest times, and those from the potential wells.
No, well, we don't have exactly this. So what we did was a little bit indirect. It was to use all this kind of mapping that we get. So we know this some sort of spatial mapping of the cellular the membrane landscape to do simulation. Because of course in this data, because we have very short trajectories, as you know, these labels are a little bit not very stable. So we don't really measure at the single protein level how long they stay. So this is kind of complementary with what we did before with brighter probes. So we have to, the way we tried to solve that was to do simulation to say if you have this type of proteins, and we get back to something which is very reasonable, at least totally compatible with what we measure independently. But I think I'm more comfortable with by separating, not using essentially the residence time as a parameter to describe. And the reason for that, it's maybe my physics bias, is that it mixes a lot of things. It mixes the fact that you can move and stay very long, the fact that going out is kind of costly in terms of energy. And so I would say as an effective parameters, it's fine, but I think that it was still a motivation to go and separate different type of contribution. So that's why we try to also follow this type of routes.
Gordon?
Yes, quickly. Do you know what happens at the level of slices? Synapses in slices.
I'm sorry, I missed. Oh, okay.
Here.
Okay. Excuse me. In slices, this experiment, I mean, I've not done this experiment, similar experiment in slices.
It's difficult.
It's still a challenge. I mean, there have been progress. I think people using slices and different type of model. The optics is more complicated. The labeling is more challenging. I would say it will come, and I think now we have to go back a little bit to the microscopy, at least for a while, to improve the imaging conditions. But now I think it was mentioned by Jan Ellenberg, all these techniques based on light sheet imaging, I think they're going to be applied because they are well designed for relatively thick sample, the type that you obtain in slice. So for the moment, I don't have the data and I cannot tell you, but I think the information will come in a reasonable future. We'll see.
Beautiful. Here, beautiful experiments. In the case of the neuromuscular junction for decades, it was known that the extracellular matrix is playing a very important role in the stability of the synapse and of the receptors as well. So here, do you have any insight on the contribution of the extracellular matrix in the stability of the synapse? And or do you view that gephyrin is the only element which contributes to the stability of the synapse?
No, I mean, then you have, I think, to be honest, I'm probably not the best person to answer all these questions. Clearly, I don't think gephyrin is the only contributor to the stability, but clearly there is this, in the case of this glycine receptor, this really relatively favorable situation for us, because then it's, to my understanding, it's largely mediated by this interaction. So now the role of extracellular matrix, this is not something I personally investigated in the experiment with Antoine's lab. Then I'm not. This is not something we. To be honest, I cannot tell you about how it would play out in this on the results here. Next time. Hopefully.
Still, I have a question. It's about adaptive optics. In astronomy, this is used to correct for turbulence. What is it used here for? I didn't quite understand.
Okay, I certainly went a bit fast on this. Okay, oh, I think I should not be too far, just almost there. Excuse me. So the problem that we have is that when we make an image of something in 3D, if I show you the movie here, we scan the position in 3D. So you see that you have these spots which gets large and then refocus and gets large again. So this doesn't give you a lot of Z information. So the role of the deformable mirror here is to do some sort of PSF engineering. So we modified the PSF such that now we encode some Z information in the defocusing. And so that's what you see in this little movie, where I do exactly the same experiment, but because I have my deformable mirror that imprint some phase profile on the light coming from the single molecules, when we move along Z, we go from something which is extended in one direction to extended in the horizontal direction. And so if we now calibrate the width along Z or X, which is the curve on the top right here, and we can, when we measure an image, we can basically just do with some sort of a lookup table, convert the asymmetry into a Z position. And the point, but this is not I mean, what you can do with the deformable in imaging is more than this. In that case, that's what it was useful for, but there are a lot of things, I think, a lot of interest of bringing this adaptive optics tool to biological imaging.